My name is Paul Mclaren. I'm a research scientist at the J.C. Welt Infectious Diseases Research Center, which is part of the Public Health Agency of Canada. So I'm a government scientist. A bit about my career path. I did my undergraduate degree in microbiology and genetics at the University of Manitoba. And after that, I went on to do a PhD in the Department of Medical Microbiology and Infectious Diseases under the supervision of Drs. Keith Folk and Dr. Frank Plummer. And that's really where I got introduced to and fell in love with doing medical research. My PhD project was focused on understanding how some individuals are able to resist HIV infection. So the University of Manitoba has a long-standing collaboration with the University of Nairobi, and they made the observation in the late 80s, early 90s, that some women who were living in HIV endemic areas were highly exposed to HIV, but remained uninfected for very long periods of time. And that's something that's been under intense study for quite a while. And my small piece of that was to look at how CD4 T cell function was different between individuals who were resistant to HIV compared to individuals who would ultimately go on to become HIV infected. And I think one of the best parts about that was number one, I had a very good experience and interaction with my advisor, my direct mentor, Keith Folk, and with Dr. Frank Plummer, who was sort of the head of that project at the time. And through that experience, I was actually able to travel to Kenya, and not only to do lab work, but also to meet and interact with the people who were impacted by HIV and who were actually on the ground, the nurses and doctors on the ground who were dealing with the HIV pandemic one-on-one. So it really gave me a personal perspective on the extent of the pandemic. I think working in the lab, you can be a little bit detached from what's going on. So I thought that was actually an amazing experience. So really, that's where I really became sort of a lifelong, or at least to this date, a lifelong HIV researcher. After my PhD, I took a fairly traditional academic route. So I went on and did a postdoctoral fellowship at the Broad Institute, Brigham and Women's Hospital, and Harvard Medical School in Boston, and I was there for 4 years, and that's where I began to study how human genetic variability influences susceptibility to infection, in particular HIV, but other infections as well as the rate of progression and response to therapy. I spent 4 years in that environment, which was a very intense period of time. Boston is a very highly motivating place, but it's also very competitive. But it was a very good experience getting different perspectives on research. So I think going away from my postdoc was very valuable. From there, I went on and did a 3-year research fellowship in Switzerland, in Lausanne, at the Swiss Federal Institutes of Technology, where I was a junior group leader in bioinformatics, and really extended my research to systems biology of HIV infection and susceptibility to infection. And after that, in 2015, I came back home to Winnipeg to work at the Public Health Agency of Canada, where my program is involved in researching how human genetic variability impacts disease susceptibility, infection susceptibility, disease progression, and response to therapy. I manage a small team of technicians and students, and really I have, as a government scientist, I have two responsibilities. Number one is to do basic research into the understanding of HIV pathogenesis and pathogenesis of infection in general, and also I have a service component. So I need to be of direct service to the Canadian people, and this is sort of achieved through reference services or development of clinically relatable research programs. So what are the positive aspects of my position? I think being involved in government science is sort of unique in that it puts you in direct contact with people from a multidisciplinary team of individuals from basic researchers to epidemiologists to people who do sort of economic impact assessments of program implementation, and then the policy and implementation people. And because I have this this service mandate, I'm actually, part of my job is to drive the results that we find in the lab all the way through to public health policy. And I'm given the contact individuals to interact with that know how to translate results from the lab all the way into public policy. So not only is that an option, but it's also part of my job. So I always need to be thinking about directly thinking about how the results that I'm generating in the lab can be translated into actual public health policy. And I think that's a very motivating factor. It's very nice at the end of the day to see something that you've worked on for a lot of years be implemented as policies. So what are the challenges of my position? The government is a big organization, so there are a lot of it can be slow to move at times. There are a lot of sort of bureaucratic hurdles that one needs to deal with from time to time in the government, simply because policies are developed for the entire agency as a whole, and even though they may not be directly applicable to you, you're still held to some parts of those standards. In addition to that, funding, although we do have a base level of funding that is quite good. Attaining additional funding from funding agencies like the Canadian Institutes for Health Research is a challenge. I mean, not just to this position, but to academic positions as well. And time management, I think, is transitioning from postdoc to actually heading my own group. I struggled early on with time management and delegation, where I needed to do a better job of allowing my projects to be driven more directly forward by people in my group, rather than spending my time doing the analysis or doing the basic science. So figuring out how to delegate and where my efforts needed to be most focused. So what are the recommendations for a trainee that would like to get a similar position? I think depending on the stage that you're at in your career, as an undergraduate student, there are opportunities to work in the federal government. We have a great program called the Federal Student Work Experience Project, or the FSWEP program. You can find it online, and this is it's actually a mechanism to connect undergraduate students with researchers in the government who have open positions for doing a research term, from a summer term to half a term off. So I think getting experience with that is great. Being involved in government research early on to see how it works, to see if the career is a good fit for you. And then beyond that, I think it really is. Most of the people who work in the public health agencies of Canada or here in Winnipeg at the National Microbiology Labs took a reasonably traditional academic route, where they've done some graduate studies, either a master's degree or a PhD, and then they've done some research fellowship work. And then, of course, in that is a responsibility to publish your results, present your results at scientific committees, at scientific meetings, and additionally, getting multiple different types of experience, I think, is good. So I had the opportunity during my PhD to be involved in a multidisciplinary training program where I was a basic researcher, but we interacted with clinician scientists, with epidemiologists, as well as social scientists. And because we're always here in the government trying to drive towards policy shifts, having some understanding of how that process works, I think, is definitely an asset to this position. And how do I see my career moving forward? We have a fairly straightforward promotion process for my position as a research scientist. That this is information that's all available online, whereas I progress year over year, I'm given slightly more resources, and I'm expected to produce some more, and then I can work my way through steps in different levels to gain more responsibility and to gain more resources, and then reclassify my position up different levels. And that's the reclassification is usually based on my sort of progress in my career academically, and how am I viewed in the field? Am I publishing good papers? Am I presenting good research at well-respected conferences? And am I meeting my service component? Are there pragmatic aspects of my work that are being applied and turned into public policy? And then this sort of reflects in the level that you can achieve through the government, and as you increase through levels, you increase responsibility, and then opportunities open up into more sort of management positions, into chief positions as well as director positions. So what is my typical work week? So as I mentioned, I manage a team of technicians and students. So in a typical work week, I like to meet, and I have a small team, so this is possible. I like to meet one-on-one with all of my team members to discuss specific aspects of their projects and provide any feedback that they need, or any additional resources that they might need access to. And I usually involved in meetings with the larger unit as well as the National Microbiology Lab as a whole, so try to stay on top of each of those projects. And as well, I like to try to save some time each week for performing my own research projects. So I do hold back small parts of the research program to actually that are my own project. That sort of keeps me sane. I got into this business because I loved research, so I like to have my own research project. And then writing papers and looking for additional funding sources. And I think one recommendation that seems to have worked for me, although it's not perfect, is to keep at least one day a week where my schedule is free from any meetings, where I actually can focus on doing a research project, or catching up on reading the papers that I've been neglecting all week, or writing a paper that I need to be publishing. So that's the average work week for me.